So gene expression regulation. We've mentioned this a number of times now, that all cells in a multicellular organism, or even all cells in unicellular organisms in a community, have the same genes. Yet, and this is particularly true of multicellular organisms, not all cells are equal. And that is because they express different genes. So they differ in their gene expression patterns. And we've mentioned a number of times that what we mean when we say gene expression patterns, we mean, it refers to where, when, and how much of a given gene is expressed. So different cells in the organism may not only not express the same genes, but also they will, even if they do share expression of some genes, they may express them in different amounts or at different times. The idea that cells not only don't express all genes, but also that they differ in their gene expression patterns leads to this concept of housekeeping versus regulated genes. And housekeeping genes, as the name indicates, are those genes that are really necessary for pretty much all cells to function, to operate. So these will include genes that play central metabolic functions in the cell, that are important for obtaining energy from nutrients, to replicate DNA, transcribe DNA, the genes like the ribosomal and transfer RNA genes in DNA that need to be expressed all the time for a cell to work, to function. So these housekeeping genes are expressed in all cells, and I have that in parentheses because it may not be applied to 100% of the cells that absolutely all the cells in a multicellular organism express them all. But most importantly, housekeeping genes tend to be expressed at all times. And then in contrast, there is the regulated genes that all cells have, but they decide to express or not depending on what cells they are, what their needs are at any given time. It may depend on the physiological state of a multicellular organism, be it an animal or plant or whatnot. So when we talk about transcriptional, or when we talk about gene expression regulation, one of the things that immediately comes to mind is transcriptional regulation. And that is the mechanisms that determine which genes are expressed, where, meaning in what cell types, when in the life of that cell, and how much of it, mRNA from a given gene, is produced. But, and this is very important, gene expression regulation is a concept that also includes these other three regulatory mechanisms. And those are post-transcriptional, translational, and post-translational regulatory mechanisms. Why? Because when we say gene expression regulation, what we really care is how the expression of a gene, or how the product of a gene, the amount of a product of a gene, is regulated in cells. And that can be done at any of these four stages. You can control how much of a product of a gene you have in a cell by deciding how much of mRNA you'll produce for it, by regulating how much of that mRNA matures into a translatable mRNA, and then how much of that mature mRNA you translate into protein, or even further, how much you modify the obtained polypeptide to really get the final functional protein coded in that gene. Okay? So these four stages, and we'll discuss transcriptional regulation today, but these are the three stages we'll discuss on Thursday, and they're equally important in determining how much of a final functional product you have of a gene in a cell at any given time. Okay, so like I said, we'll talk about transcriptional regulation today. In other words, how much mRNA is produced from a coding gene. And we'll split the lecture in two. We'll talk about prokaryotic transcriptional regulation first, and then eukaryotic transcriptional regulation. And we'll start with prokaryotes. So prokaryotes, we've said this also a number of times, prokaryotes are in many ways all about economy. And one of the things, or one of the ways in which this drive for being economical in all of their biology manifests itself is in the way they regulate the transcription of their genes, of their regulated genes. So whereas the housekeeping genes, as I was saying before, are pretty much expressed all the time, there is these regulated genes that bacteria may not, or prokaryotes may not need all the time, and they need to make a decision of when they should turn them on or not. So what prokaryotes do is they tend to organize their regulated genes in operons. And operons are really clusters of genes that are all needed for a specific function. So they're all involved in the same function. So in this example, we have genes 1 through 5. This is a generic example. And these 5 genes will all be involved in a process. So it could be that they're involved in 5 different steps of a sequence. It can be that they are all needed to form a 5-protein complex that is needed for a process or function. It could be in a number of ways that these 5 genes are involved in the same process. But the point is that when the cell needs one, it will need all 5 of them. So it will regulate the transcription of all 5 at once. Turn their expression on or off in concert. Now, what's interesting about operons, prokaryotic operons, and there's very few examples in eukaryotes of this, very few. So it's mostly a prokaryote thing, is that these clusters of genes express a single polycistronic mRNA. So there's going to be one mRNA molecule there that will carry the information to translate 5 different polypeptides. Now, when we were talking about translation from mRNAs in prokaryotes, we mentioned that the ribosome knows to bind an mRNA, a prokaryotic mRNA, through a sequence called the Shine-Dalgarno sequence. And that's where it will start scanning for the AUG, and that's where translation will begin. When we were talking about that example, we were talking about a monocistronic mRNA, where an mRNA molecule would give rise to one polypeptide. Now here we're dealing with 5 different polypeptides coded in the same mRNA molecule. So the ribosomes will need a way to know that they have to enter this mRNA molecule at 5 different spots. Here, 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 and there, and there. Well, that didn't really leave room for much of a polypeptide. Okay? So the idea here is that you have these 5 entry points for the ribosome such that 5 different polypeptides can be translated from this polycistronic mRNA. Okay? You may come across this concept as the IRES, and that stands for Internal Ribosomal Entry Site. And they may come with different names too. But once again, this is much more common in prokaryotes, not so much in eukaryotes, though it does exist in eukaryotes as well. And the idea is that you code a little sequence in your mRNA such that you let know the ribosomes in a cell that there is various entry points for them such that they can start translating the various polypeptides coded in a polycistronic mRNA. So in a way, not only is the prokaryotic cell transcribing all of the genes at once, it's translating them all at once too, adding another layer of coordination in the expression of all of these genes that are needed for the same process. In an operon, you have 2 components. You have the actual transcriptional unit that gets transcribed into these polycistronic mRNAs, and you also have the regulatory DNA sequences. And those are in this schematic, the promoter and operator sequences that sit upstream of the transcriptional unit, and those are the ones that indicate to the rest of the cell. They give the instructions, they contain the instructions for indicating to the rest of the cell when to transcribe that polycistronic mRNA or not. Okay, so that's a very generic description of an operon. The polycistronic or the transcriptional unit that will code for polycistronic mRNA plus the DNA regulatory sequences, in its simplest form, a promoter and an operator sequence. So the most textbooks, and your book is no exception, will discuss the lac operon first, and we'll do that in a few minutes. And then the trip operon, and there is historical reasons for that. The lac operon was the first to be described, and it's a little more complex. I personally like starting with the discussion of the trip operon because it's a little simpler and easier to understand. So we'll discuss these first, and then we'll talk about the lac operon. So the trip operon is no different from that generic description we had of an operon in that the trip operon has these 5 genes named trip E through A, backwards, E, D, C, B, A. And what the proteins encoded by these genes do is to synthesize tryptophan. These are the enzymes that will take a bunch of biomolecules and make tryptophan out of them. So they encode the enzymes for the tryptophan biosynthesis pathway. I just said that the DNA regulatory sequences of an operon encode for the logic of that operon. And what I meant by that is the question that a cell asks, or a series of questions that a cell asks to decide if it needs to turn on the operon or not. And in this particular case, for the trip operon, the question that the cell asks is, do we have any tryptophan? If there is tryptophan inside a cell, there may not be a need to make more. So the cell may decide to turn off the expression of these genes. Remember, expression of genes is a fairly costly process, and bacteria and prokaryotes are all about economy. So if the bacteria already have tryptophan, why make the enzymes that you need to make more? So if there is tryptophan, the operon remains off. But if there is no tryptophan, then let's make it. Let's turn on this operon, let's make the enzymes that we need to make tryptophan. So if there is no tryptophan around, RNA polymerase will bind the promoter sequence of this operon and start the transcription of this polycistronic mRNA that will lead to the translation of the 5 enzymes needed for tryptophan biosynthesis. So far nothing new under the sun. This is typical transcription translation. Not any different except for this polycistronic thing from what you've seen before. On the other hand, if there is tryptophan, why bother? Why waste our resources making enzymes to make something that we already have? So the cells turn the operon off. So we say, because given the condition of tryptophan around, the operon is turned off, we say that the tryptophan operon is a repressible operon. The cell has the ability to turn it off. And it does so using a protein called the tryptophan repressor. Go figure. That as a repressor becomes active when it binds tryptophan. So the logic here is, if there is a lot of tryptophan in a cell, that tryptophan will bind to this tryptophan repressor and activate it through the usual, some sort of conformational change that this protein undergoes such that it balances the electrochemical profile in its structure, And through that conformational change, it can now bind DNA through the operator sequence and block RNA polymerase. So now RNA polymerase cannot transcribe the operon because the tryptophan repressor is there, active, blocking transcription. Now, it follows that the situation we were describing before, when there is no tryptophan around, that is because the tryptophan repressor, in the absence of tryptophan, is inactive. Without tryptophan bound to it, it undergoes some sort of conformational change that doesn't allow it to bind DNA through the operator sequence. And without it in the operator sequence, RNA polymerase can bind the promoter sequence and transcribe the operon just fine. One thing to notice here is that the tryptophan repressor is a protein itself, so it has to be coded by a gene somewhere. And that's exactly the case. There is a tryptophan R or tryptophan repressor gene somewhere else outside of this operon. It doesn't have anything to do with the operon per se, which will be transcribed into an mRNA that is translated into the TRP repressor protein itself. Okay? So that's a relatively, it's about the simplest that an operon can get. There is the transcriptional unit, the promoter sequence, and what establishes if that transcriptional unit is transcribed or not is the presence or absence of this repressor protein that will block the transcription by RNA polymerase. And the presence or absence of this repressor will depend on a condition. In this case, is the tryptophan in this cell. And there's many, many operons that operate that way. The cell asks a question. Do I need X, tryptophan, or some other amino acid, or some other biomolecule? And if I don't, because there is plenty of it around me, so then there is some repressor protein that blocks the transcription of that operon. That's sort of the case with all of the repressible operons. Then the slightly more complex case, it begins very similar, the lac operon, but then it gets a little more complex. So let's talk about the lac operon now. The lac operon, or lactose operon, has 3 genes. And these 3 genes are involved in consuming lactose. So one of them will help lactose into the cell, another one will start processing it. Bottom line is these 3 genes are involved in extracting energy from lactose, okay? Which is a sugar, one of these disaccharides made of 2 sugar monomers. So the logic in this operon goes more or less like this. If there is no lactose around, and here around I mean more literally, if there is no lactose outside of the cell, and hence inside, why bother? Why making these enzymes that are for extracting energy from lactose if there is no lactose available? So here, the absence of lactose turns the operon off. Before it was the presence of tryptophan that turned it off. But here it is the absence of lactose that turns it off. And again, that is because of a lac repressor protein that, unlike the TRP repressor, is active when it's empty. So here, the empty repressor has the right 3D conformation to bind the operator sequence in DNA and block transcription of the operon. Now, if lactose shows up, both in the outside and the levels of lactose start building up in the inside of the cell, and by the way, there is a sort of a surrogate molecule that the cell really uses to measure levels of lactose. This allolactose molecule is a derivative of lactose that is the one that truly does the sensing for the cell. So if there starts to be more lactose, and hence allolactose in the cell, so then the cell decides that it might be a good idea to turn the operon on to express the enzymes that can extract energy out of lactose. And once again, the way it works is kind of converse It's the opposite or reverse from what happens with the trip operon, because you have the allolactose binding the repressor and inducing that conformational change that will get it off of DNA. So when the lac repressor is occupied by allolactose, it cannot bind DNA, and now RNA polymerase can proceed with transcribing the 3 genes in the operon. There will be this polycistronic mRNA that will be translated into the 3 enzymes that are needed for lactose catabolism, for extracting energy out of lactose. Because the presence of lactose will lead to the turning on of this operon, we say that the lac operon is an inducible operon. So as you can see, it's essentially the mirror image of what was going on with the trip operon. But in both cases, the cell asks a question. Do I need to transcribe these genes? And the way it molecularly asks that question is by the conformation of these repressor proteins that will allow them or not to bind the operator sequence in DNA and hence block or not the transcription by RNA pol. So, is that it? Is that how all operons function? Is it that simple? A cell just decides what genes it wants and then turns the operons on or off with a repressor? Is it that simple? Not quite so, yeah. It's usually a few layers of complexity added to this problem. So what we've just discussed in a way is an example of either, so there are some genes that are permanently in a stop. And those are genes that a cell, for whatever reason, decides that it doesn't need to make, period. This is probably more applicable to multicellular organisms where you have cells that don't need to make a given protein ever. So why would a muscle cell make a protein that is important for detecting light? It doesn't need it. That's something that retina cells need, not a muscle cell. So it won't ever make that protein. So it just puts a stop on it, period. Then these operons that we were just discussing, in a way, are example of more complex signals in which there is a decision to be made. There is a question being asked and a decision to be made as to whether or not to express a given operand. And speaking of traffic lights, there are cases, and the lac operand is an example of that, in which it's not only about a red light, it's not only about stopping, you also need, at some point, the green light to go on. And that's exactly what happens with the lac operand. We'll discuss that next. So up to now, I just simplified it a little bit. I said the cell asks, is there lactose around? If there is lactose, yeah, let's make those enzymes because we can extract energy out of that lactose. But there is another layer of complexity or regulation, and it has to do with the presence of glucose in the cell. So a bacterial cell won't only ask, is there or not lactose around? It will also ask, okay, but what about glucose? And why do you think a cell might care about whether or not there is glucose around?
It's a monosaccharide, it's easier to use.
It's a little easier to use, and it's the one that kickstarts glycolysis. So it's sort of a couple of steps forward in this process. So if there is a lot of lactose, sure enough, you can extract energy out of it. But if there is also a lot of glucose, in a way, we're going again into this mode of a wasteful process, because you'd be processing this lactose, you'd be expressing all these genes that you need to extract energy from lactose, when in reality, you're bathing in glucose, and you could extract all the energy you need from glucose as well. So the cell asks two questions. Not only how much lactose is there around, but how much glucose is there around. And the logic goes like this. If there is lactose, but there is also a lot of glucose, why bother? For the reasons that we just discussed. Now, on the other hand, if there is a lot of lactose and no glucose, then yes, let's turn on these genes because we can extract energy out of this. There is no other source of energy that we can get our hands on, so let's use lactose, let's express these genes. So when it comes to the lactose side of things, we already talked about it. There is the LAC-I for LAC inhibitor gene that expresses the LAC repressor that is active in the absence of allolactose but becomes repressed in the presence of allolactose. So if there is a lot of lactose around, the repressor will be off of the operator sequence, and it would allow RNA polymerase to transcribe the operon. But then there is the second question, is there glucose around? And only if the answer to that question is no, will RNA polymerase transcribe these genes. And how does RNA polymerase know, or how does the cell know? Well, it turns out that when there is a lot of glucose in a cell, there will be a lot of ATP. Do we all agree on that? Yeah? Lots of glucose, lots of glycolysis, lots of oxidative phosphorylation, whatnot, lots of ATP. Now, when there is very little glucose or no glucose whatsoever, that ATP starts being consumed. It's first turned into ADP in many reactions that will extract energy out of the hydrolysis of ATP to ADP. And then there are some reactions that can extract further energy from going ATP to AMP directly or ADP to AMP. Point is that when you have very little glucose, you will have lots of AMP. There is no energy to turn that AMP back into an ATP, and the levels of AMP start building up. And when the levels of AMP start building up, there is this little reaction that happens within the cell that will turn AMP into CAMP for cyclic AMP. Okay? Cyclic AMP is an intramolecular covalent bond in the AMP molecule that will turn a linear molecule into a ring. And that cyclic AMP can activate this other protein called CAP. And when CAP is activated because it bound CAMP, cyclic AMP, it will go and bind DNA and recruit RNA polymerase. So here's the full logic of the LAC operon. In the presence of lactose, the lactose repressor is inactive and off of the DNA. So RNA polymerase, in principle, could bind and transcribe the operon. But it will do so much more efficiently if there is a lot of CAP, activated CAP bound to DNA to recruit it to DNA to initiate transcription. And that is a consequence of there being very little glucose within those cells, which will raise the levels of cyclic AMP, which will activate the CAP protein, and which will allow it to bind DNA. Now, once you understand the molecular functioning of the LAC operon, it's rather easy to understand why the various conditions will dictate whether a cell turns the operon on or not. So, for instance, if you have very little glucose and very little lactose, the very little glucose will lead to very high levels of activated CAP bound to its CAMP. So you would imagine, oh, great, lots of activated CAP, lots of RNA pol transcribing. The LAC operon is turned on. But don't forget that if there is very little lactose, the lactose repressor becomes active because it's empty, and now it can go bind the operator sequence and block that RNA pol. So the logic of the lac operon is dual. It's not only about lactose, it's not about glucose only, it's a combination of the two. The perfect condition for the lac operon to be expressed at its maximum is a lot of lactose and little or no glucose. And the worst condition possible is a lot of glucose, which will inactivate the cap protein and get it off of DNA, and no lactose. That's when the cell really thinks, why bother with this operon? We have all the glucose we need, we don't have any lactose to process, why turn on these genes? So, any questions about either operon, operons in general?
You see, the best way is when there has a lot of lactose and very few glucose for the process to occur.
Yeah, because that's when the lac repressor being bound by lactose is inactive, as shown here. So when the lac repressor has allolactose bound to it, it's inactive, so it cannot bind the operator. So it's off of the DNA and it won't block the RNA polymerase. And then on the other hand, having very little to no glucose, all of the ATP in the cell will slowly turn into first ADP and then AMP. And once you have lots of AMP, you're going to start building up the levels of cyclic AMP. And cyclic AMP is the one that binds the cap protein and activates it, allowing it to bind DNA. Now, the lac repressor will block RNA polymerase. The activated cap protein will actually recruit it to DNA. So these two proteins are binding DNA in the lac operon, but they're doing two different things. One would be blocking the activity of RNA polymerase. The other one is actually inducing it. Okay. Any other questions? All right, so let's talk about eukaryotic genes now. Let me start off by saying that eukaryotic genes are usually not organized in operons, but that is not to say that their expression cannot be coordinated. So, like in prokaryotes, there are many eukaryotic genes that work in concert. And it is in the best cell's interest to express those genes in a coordinated manner, such that when you need one, you express all the ones that you need, and when you don't need them anymore, you turn off all of their expressions. But even though that still holds for eukaryotic cells usually don't coordinate the expression of these groups of genes in operons. We'll discuss a little bit later how they do it. Now, let's talk about the expression of a eukaryotic gene first, and then we'll talk about the coordinated expression of a group of them. If you remember from when we were discussing the organization of genes, the typical eukaryotic gene has a core promoter. That's a sequence in DNA that in some cases includes the TATA box, but doesn't have to always. There are some core promoters that are called TATA-less, meaning that they have other special sequences, but not the TATA box. And the core promoter, what it does, its job is recruiting RNA polymerase, period. That's where RNA polymerase will bind DNA and start transcribing that transcriptional unit. What we didn't discuss in much detail, even though we mentioned it briefly, is the proximal promoter. Proximal promoter is also a sequence in DNA that is just upstream from the core promoter. And it's not the place where RNA polymerase binds and initiates transcription, but it's the place where these regulatory proteins bind DNA to stimulate or inhibit the transcription of that transcriptional unit. Okay? So it's the place where these transcriptional activators and inhibitors bind DNA and induce or inhibit the expression of a gene. And finally, there is enhancers. And enhancers are, in essence, in their function, they're not much different from a proximal promoter. So these two are very similar. The difference is that an enhancer is far away from that core promoter. So the proximal promoter, as the name indicates, is proximal to the core promoter. But enhancers operate at a distance. They're further away. Now, here's an important thing. They can be upstream or downstream of a gene. In many diagrams, you'll see enhancers being far away from the transcriptional unit that they regulate in the upstream direction, like in this diagram here. Right? So you have your transcriptional unit here, and then the enhancer is sitting some way upstream of the gene. But enhancers can be anywhere. They can be upstream, downstream, or sometimes even in the middle of a gene. What defines an enhancer is a sequence in DNA that is not right next to the core promoter and that binds a bunch of transcriptional activators and inhibitors that can regulate the expression of a transcriptional unit. Okay? So, we've just defined the components of a gene from a DNA point of view. There is the transcriptional unit and then the core promoter that binds RNA pol, the proximal promoter, and the enhancers. Now let's talk about what happens from the protein side. To transcribe a transcriptional unit, ultimately what you need is RNA polymerase transcribing that DNA. But before RNA polymerase can bind to a transcriptional unit and start transcribing it, you need a whole bunch of general transcription factors that assemble at the core promoter and allow for the recruitment of RNA polymerase. So far we've simplified it a little bit and we made it sound like RNA polymerase will bind to DNA and start transcription. It's slightly more complex. RNA polymerase will bind to DNA only if there is this assembly of general transcription factors at a core promoter already in place. So that's what happens at the core promoter. What about these transcriptional activators and inhibitors that I mentioned before, or repressors that I mentioned before? They can operate either in proximal promoters or in enhancers. So what they do is they either function not so much by recruiting RNA polymerase or inhibiting the recruitment of RNA polymerase to DNA, but by acting on these general transcription factors that we just mentioned. So an activator can activate transcription by bringing more of these general transcription factors to a core promoter. And an inhibitor can do the opposite. So they don't work directly on RNA polymerase. They work on the general transcription factors that will bring RNA polymerase to the gene. So there is this indirect effect on the rate of RNA polymerase binding to DNA. Another way that they have to operate, and we'll discuss this a bit more on Thursday, is instead of working on the general transcription factors, they work on the DNA itself, where the general transcription factors would bind. So instead of interacting directly with the general transcription factors, what they do is they modify DNA in the core promoter sequences such that they indirectly affect the binding of these general factors to the promoter. So here, the action on the general transcription factors is indirect. First, there is this modification of DNA, and that affects the binding, or lack thereof, of the general transcription factors. And by the way, as I mentioned before, enhancers do the exact same thing. Enhancers regulate the binding to DNA of these transcriptional activators or repressors, and they can work also either directly by regulating the activity of the general factors or through modifications of DNA, chromatin modifications, which from now on you'll spell modifictions. New word. Better learn it, modifictions. So this is a schematic that shows what I just said. The idea is in a core promoter, you have, in some core promoters, you have your tata box, that sequence that is bound by the tata box binding protein or TBP. The binding of the TBP to the tata box will trigger the recruitment an assembly of all of these general transcription factors. And at some point, when enough of them have assembled and they have all of the right conformations and whatnot, they bring RNA polymerase to the DNA and transcription begins. Now, these proximal promoter sequences that sit just upstream of a transcription or of a core promoter, or the enhancers that sit somewhere else, the way they work is by bringing these transcription factors, either activating or inhibitory, that will directly or indirectly either induce the assembly of the general transcription factors or inhibit it. And once again, I say directly or indirectly because they can do so by interacting with these general factors at a protein-to-protein level. So there's a molecular interaction between the proteins themselves, or indirectly through modifications to the core promoter sequence that will in turn affect the way these general factors bind to it. And enhancers are not much different, except that they operate at a further distance. So in an enhancer, you will have these, say, activator proteins that can bring some of these general transcription factors to the core promoter of a gene. So the idea here is you had a core promoter with some activators bound to it, but that wasn't quite enough to bring all of the general factors and RNA polymerase that is needed to initiate transcription. Now, because of the activator proteins bound to the enhancer further away, and because of something that we know as DNA looping, these activator proteins bound to the enhancer would form a complex of proteins along with activators in the core promoter sequence that, as a whole, will much further induce the assembly of the core factors in the core promoter, or the basic factors and general factors in the core promoter. And again, this is going a little too molecular, but just so that you guys understand the full picture, it's always the same. It's all about rates of binding and separation between molecules. So the fact they have all of these proteins, these activators bound to DNA, and bound to proteins that don't directly bind to DNA, but that form these very large protein complexes, all they do is indirectly increase the rate of binding of these general factors to the core promoter. The general factors could bind the core promoter by themselves, possibly, but the presence of all of these proteins in enhancers and proximal promoters, what they do is they increase the rate of that happening. So when all of these things are in place, the basic factors will bind to the core promoter more often, which will lead to a more frequent recruitment of RNA polymerase II to that transcriptional unit, and hence to the transcription of that gene. So I started off this section saying that that eukaryotic genes are usually not organized in operons. That is not to say that they don't respond to a regulatory logic like operons do. When we were talking about operons, we were talking about these repressor proteins, and in the case of the lac operon, we were talking about the cap protein. With eukaryotic genes, something very similar goes on. there is all of these activator proteins and all these repressor proteins. And it is sort of the combinatorial action of these repressors and activators which decides when a gene is transcribed. So let's suppose that under a given condition, in this particular cell, gene X here has a bunch of repressors bound to DNA, repressors 1 and 2. That gene won't be transcribed because there is those repressors somehow blocking the activity of RNA polymerase, possibly by not letting the general transcription factors bind the core promoter and so forth. Now, something may happen to that cell that changes the presence of those repressors by the presence of now a bunch of activators. And like in the more complex operons in prokaryotes, eukaryotes can follow fairly complex logics. So it's not always presence or absence of one repressor. It may be multiple repressors. And it's not always the presence or absence of one activator. It can be multiple activators. And each of them will respond to a given question. So it could be, for instance, back to our example with the muscle cell. A muscle cell may need to decide if it needs to express myosin, which is one of the main proteins involved in a muscle cell contraction. Okay? So in principle, you would imagine, well, why wouldn't a muscle cell want to express myosin? It will always want to express myosin. Maybe not. A mature muscle cell that is fully functional doesn't need to express anymore. Anymore would be an excess. It may actually become a little detrimental to that muscle cell. So at some point, if the cell can sense that it's working properly and that it's contracting just fine, it may decide to turn off the expression of that myosin gene. And that will be through, say, repressor 2. Repressor 1 may be a completely different protein that tells a muscle cell not to express myosin because, for instance, that muscle is not being used much. And in fact, that muscle cell needs to regress a little bit. There's no need for so many myosin fibers, so it actually needs to get rid of myosin. So that may be through a different repressor, repressor 1. And then in a different cell type, say a retina cell, there could be yet another repressor, repressor 3, that functions by saying, hey, we're not a muscle cell, we shouldn't express any myosin. Okay? So these different repressors will respond to different questions that a cell can ask. And the same goes with the activator. Back to our muscle cell, activator J might say, we need more myosin because the ones that we had, even though it was just enough, the ones that we had are starting to break a little bit, so we need to replace it. Activator I may respond to a signal that says, this muscle is being used a lot, and we're not really coping with the need for these muscles. So we need to step it up, we need to grow, we need to make more myosin because we'll have to grow this muscle. And then activator N could respond to a different question asked in that muscle cell. So each of these will combinatorially operate on the same gene. And in a way, their presence or absence is really a series of answers to a series of questions that a cell asks before it decides to turn on the expression of a gene or not. So for a gene to be expressed, you often need multiple activators being present, multiple repressors being lifted off or absent, and then finally, the proper chromatin conditions, because this has to be an area of DNA that is not silenced, that is accessible to all of these factors and to RNA pol and whatnot. So, an example of everything that we just said is a decision that a cell needs to make at some point to divide or not. And that decision will depend on a whole bunch of signals, not only from the inside, but also from the outside. So for instance, you have all of these growth factors that stimulate cell division and all these growth inhibitors that inhibit cell division. And the way they work is there are all these signals that might come to a cell and instruct it to divide. Those signals, as we will discuss next week, will be detected by cell surface receptors. And when that happens, there is a signal transduction pathway. I would give you a hard time if I hear all that beeping from your phone, so I should do the same. So these signals bind cell surface receptors, and there is a signal transduction pathway that will ultimately activate or inhibit these transcriptional factors, these TFs in the nucleus. So next week we'll discuss signal transduction pathways, and we're not really going to discuss much of the end of the story. The end of the story we're talking about today. All these signal transduction pathways that sense things on the outside of the cell and communicate the information to the inside of the cell ultimately regulate the activity of these transcription factors. They do other things, of course, but one of the main things they do is regulate the activity of these transcription factors. So a growth factor, for instance, might activate a transcription factor that is important for making a protein that stimulates cell division. Now, a way a cell has to inhibit cell division, or a signal has to inhibit the division of that cell, is by having a similar signal transduction pathway that activates a repressor for the same gene. So here's an example in which you have growth factors stimulating the division of that cell and growth inhibitors trying to stop it. And the way they battle it out is at the gene level by having some activators present but also some repressors. And possibly the overall outcome of this combination of activators and repressors is the gene will not be turned on because the repressors are there, and even though the activators are there, so are the repressors, so the gene remains off. So next week, when we talk about signal transduction pathways, at some point I'll say, and of course these pathways will lead to changes in the regulation of gene expression. This is what we mean. There will be these cascades of molecular events that may lead to the activation or the inactivation of these transcription factors. And at the end of the day, it's the combination of activators and repressors that will determine if a transcriptional unit in a eukaryotic cell will transcribe or not. Yes?
Next week or Thursday?
Next week. On Thursday, we'll talk about some those extra mechanisms for regulation of gene expression that have to do with what cells do to the mRNAs to make them mature or to translate them. So, any questions about gene expression regulation? No? All right, so why don't let's go through some of these. The first one, 94% of you got wrong. What? So most of you said that this is true, but it's false. Why?
It's also inhibits.
It's also what?
It also inhibits.
That's the way to look at it. No, what I had in mind is that there is these other mechanisms that ultimately lead to regulation of gene expression. So it's not only about regulating the transcription of a gene, it's also about regulating the processing of the pre-mRNA in eukaryotes, and then the regulation of translation and post-translational modifications made to a protein. So, in a way, when you take two cells and one cell has more of a protein than another, doesn't necessarily mean that the first cell transcribed more of that gene. They may have as well transcribed the same amounts of the same gene, and yet one of them ends up with more protein because the mRNA is made more stable, or because it is translated more, or because once the protein is translated, again, it's made more stable through post-translational modifications. And we'll discuss all of that on Thursday. Enhancer sequences can be shared by different loci. And a slight majority of you said that this is true, and that's correct. It is true. And we didn't discuss it in detail, but it's the time now. This is how eukaryotic cells coordinate the expression of several genes that are involved in the same process or function. In prokaryotes, all of those genes were organized in a cluster that makes an operon. In eukaryotes, what happens is the same enhancer that will use the same logic to respond to a series of regulatory questions will regulate the expression of several loci at once. So the way that looks is you have your DNA and you have an enhancer sequence here, and then you have these different loci which we'll represent with these arrows that indicate initiations of transcription. And this enhancer will coordinately regulate the expression of all of these loci at once, because enhancers work through DNA looping and they can be anywhere with respect to the transcriptional unit that they regulate. So there may be instances in which the enhancer loops that way and regulates the expression of these two guys, and then it will turn to the other way, loop in the other direction, and regulate the expression of those other two. Okay? So it is very common that a given enhancer will regulate the expression of several loci. And that's how eukaryotes regulate the expression of several genes at once. What about the third one? For the third one, it was a perfect 50-50 split. So transcription factors need to be bound to DNA to regulate expression. Who thinks that's false?
Isn't it false because there's inhibitors that don't necessarily bind to DNA itself, it just sends signals to operant?
Or activators. So either way, both activators and inhibitors can regulate transcription without binding to DNA. And remember, there were those examples in which you had your core promoter here and your basic factors here, and then your RNA pol ready to initiate transcription. And then there were all of these multi-protein complexes that often depended on the presence of an activated enhancer somewhere else. Remember? And all those proteins in those multi-protein complexes, most of them don't bind DNA. They're there to recruit the basic factors to the core promoter. They're there to stabilize the binding between them and the RNA pol, say. But they don't bind DNA directly. They know where to go thanks to these other transcription factors that do bind DNA in enhancers or in proximal promoters. So transcription factors don't need to be bound to DNA to regulate expression. This one, quite the opposite. Almost all of you got right. It is true that, like protein coding genes, some enhancer sequences can be highly conserved across species. So that is true. And the explanation for that is rather simple. These sequences in enhancers need to be such that are recognized by proteins that need to know exactly where to bind or where not to. So even though these proteins are non-coding, they're not going to be translated into a polypeptide sequence. They need to be very precise. Changes to these enhancer sequences can make an activator protein not recognize them anymore and therefore not bind there anymore, or a repressor not recognize them and not bind there anymore. And when you have an activator or a repressor that doesn't bind to a proper enhancer anymore, you will deregulate the expression of a target gene, and that can have very serious consequences for a cell. Therefore, evolution makes sure that the sequence of these enhancers remains fairly stable, that doesn't change much. Any deviations in the sequence of those enhancers will be likely selected against. And finally, this is sort of a tricky one, and no surprise, although a slight majority of you got it right. So, in addition to mRNAs, the other two important molecules involved in gene expression regulation are tRNAs and ribosomal RNAs, and that is false. They're important for making proteins, sure enough, but they don't have any role in regulating the levels of expression of a gene. They will make the proteins, granted, but they're not the ones, as we will see on Thursday, despite of all of these other processes by which cells control the amount of gene product in a cell. These two are usually not involved in any of that. All they do is they sit there and they wait for a mature mRNA to go translate. That's it. But everything else that goes into determining the levels of mature mRNA that happens earlier, or everything that happens to proteins after they've translated it, it's totally out of their control. So typically, tRNAs and ribosomal RNAs are not regulatory. They just do their job, and it is these other mechanisms that dictate how much of a gene is expressed. All right, that's all. So I'll see you on Thursday.